Hello friends, today we are going to study the gel electrophoresis. In that we will study the Agarose Gel Electrophoresis. So first of all, what is the electrophoresis? The electrophoresis is the migration of charged particle under the influence of electric field. Means electro means the charged particle and the phoresis means moment, and the many important biological molecule such as amino acid, peptide, protein, nucleotide, nucleic acid possess ionizable group and therefore at any given pH, and on the basis of under the charge of an electric field, these charged particles will migrate either to cathode or anode depending on the nature of their net charge, and this is one of the most fundamental processes used in all type of molecular biology. And now, what is the definition of electrophoresis? The definition of electrophoresis, it is the migration of charged particle or molecule in the medium under the influence of the electric field. And what is the gel electrophoresis? It is the type of electrophoresis. The gel electrophoresis involve the use of gel as a supporting media for the separation of DNA, RNA or protein under the influence of electric charge. It is usually performed for the analytical purpose but may be used for the preparative technique or partially purify molecule prior to use for other method such as the mass spectrometry, PCR, cloning DNA, sequencing and immunoglutting. And so what is the principle of gel electrophoresis? The it is the electromotive force generated across the electrode pushes or pull the molecule nucleic acid or protein through the gel matrix. The molecule moves toward the anode if negative charge or toward the cathode if positively charged. And a typical gel electrophoresis apparatus, it is the two kind of gel electrophoresis: vertical gel apparatus and horizontal gel apparatus. The vertical, it is used for the separation of the protein in SDS and PAGE, and the horizontal gel apparatus, it is used for the amino electrophoresis and electrophoretic, focusing on the electrophoresis of the DNA and RNA in agarose gel. So the different type of gel electrophoresis, in that agarose gel electrophoresis and SDS-PAGE means the sodium dialkyl sulfate and polyacrylamide gel electrophoresis, the pulse field gel electrophoresis, the two-dimensional electrophoresis. So the gel electrophoresis, in that the agarose gel electrophoresis and SDS-PAGE, and it is used for agarose gel electrophoresis is generally used to separate the large pore size like DNA and RNA, and the polyacrylamide gel electrophoresis, which is used to separate the small pore size like example protein, small size DNA. The type of gel electrophoresis, in that the agarose gel electrophoresis. Now study the supporting media. Agarose gel electrophoresis, the supporting media in this type of electrophoresis is agarose gel. This is used for the electrophoresis of nucleic acid like DNA and RNA. So what is the principle of agarose gel electrophoresis? When the partial difference is applied across the electrode of horizontal electrophoretic tank containing agarose gel, and biomolecule such as nucleic acid are loaded and they get separated according to their molecular size. The bigger molecule have more molecular size and smaller molecule have small molecular size, and the move to their respective electrodes. Here the agarose gel act as a sieve. As a sieve, the larger particle stay above and particle which are smaller than the pore size passes through it, and similarly in the gel the larger and bulky molecule stay behind, whereas the smaller molecule move faster and quickly toward their respective electrode. This process may be imagine like the running competition, the one who is thinner and have the flexible body, which will be at the ending point sooner than the one who is flat and bulky. So the instrumentation of that, the different parts is used in that, the physical apparatus and chemical component. In the physical apparatus, in that the following three types, and it consists of the electrophoretic apparatus. The horizontal gel electrophoresis system is designed for the very fast and clear separation of DNA restriction. The fragment in agarose gel, the gel apparatus vary according to manufacturer. This is the simplest apparatus is called the flat bed horizontal apparatus, and this apparatus consists of the two buffer chamber, a weak port from the agarose and flat horizontal bed of agarose upon which a small well for sample loading are located. The power supply. For the standard agarose gel electrophoresis, a voltage of 5 volt per centimeter of the gel is applied, and the centimeter value is distance between the two electrode, not the length of the gel. And third is the trans illuminator. This is the UV light box which is used to visualize ethidium bromide stained DNA in the gel. The chemical component. Second, the chemical component. This includes all the chemical components which are involved in the electrophoresis of the biomolecule. In that, different supporting medias are used. The agarose is the polysaccharide extracted from the seaweed. It is linear polymer composed of the alternating isomer of the sugar D and L galactose. The agarose melt approximately at 90 degree and the gel approximately at 40 degree. This gelatin result is in a mesh of channel with the diameter varying from 50 to 20 nanometer. The diameter of these channel determine the final porosity of the gel and act as sieve. By using the gel with the different concentration of the agarose, one can resolve different sizes of DNA fragment. A higher concentration of agarose facilitate separation of the small DNA, while the low agarose concentration is maintained during the separation of DNA having higher molecular weight, and it is the typical used at the concentration of 0.5 to 2%. Second, buffer. The gel is immersed within a electrophoresis buffer that provide ion to carry the current and some type of buffer to maintain the pH at relatively constant value. Depending on the size of DNA electrophoresis and applications, the different buffer can be used. For agarose electrophoresis, the TAE buffer or the triacetate EDTA is the most commonly used agarose gel electrophoresis buffer. And what is the TAE? Has the lowest buffering capacity of the buffers. However, the TAE offer the best resolution for the higher DNA, and TAE also require the low voltage and more time. And the TBE buffer, the triborate EDTA, is oftenly used for the smaller DNA fragment, that is the less than 500 BP. Sodium borate or sodium borate is the new buffer, but is ineffectively for the resolving fragment higher than the 5 kb. The sodium borate has advantages in its low conductivity, allowing the higher voltage up to 35 voltage per centimeter. This could allow the shorter analysis time for the routing electrophoresis. The two types of buffers are used in the process of agarose gel electrophoresis. These two types, in that this type of buffer is used Both the gel placed horizontal tank, usually triacetate, or the triaceborate, is taken electrophoresis buffer. Loading the buffer, this contain a dense medium, example the glycerol, to allow the sample to fall into the sample well, and one or two tracking dyes which migrate in the gel and allow the visual monitoring of the extent of the electrophoresis. So why dye is used? Third, the dye. Why dye is used? This is the tracking agent. This is the tracking agent that aim at visualizing the separated nucleic acid after the process of the electrophoresis. In the agarose gel electrophoresis, ethidium bromide is used as dye. The ethidium bromide is the fluorescent dye that bind to DNA and intercalates between the stacked bases. When the irradiated with the UV light of wavelength of 302 nanometer, ethidium bromide will emit fluorescent light of the wavelength 590 nanometer, that is orange in color. After the process of electrophoresis, the gel is observed under the beam of UV light produced by the trans illuminator. The dye is added to the gel during its preparation only. So what is the procedure of that? The first of all, agarose powder is mixed with the electrophoresis buffer to desired concentration, then heated in microwave oven until the completely melt. The most commonly ethidium bromide is added to gel. Final concentration 0.5 to mg per ml. At this point, the facilitate visualizing of the DNA after the electrophoresis. After cooling the solution to above about 60 degree, and it is poured into the casting tray containing a sample comb and allowed to solidify at room temperature. After the gel has solidified, comb is removed using care not to rip the bottom of the well. The gel is still in its plastic tray is inserted horizontally into the electrophoresis chamber and just covered with the buffer. The sample containing DNA mixed with loading buffer are then pipetted into the sample well, and lead and power lead are placed on the apparatus and the current is applied. It should be confirmed that the current is flowing by the observing bubble coming off the electrodes. The DNA will migrate toward the positively electrode, which is usually colored red. When the adequate migration has occurred, the DNA fragments are visualized by staining with the ethidium bromide. This fluorescent dye intercalate between the bases of DNA and RNA. This is often incorporated into the gel so that staining occurs during the electrophoresis, but the gel can also be stained after the electrophoresis by soaking in the dilute solution of ethidium bromide. To visualize DNA or RNA, the gel is placed on UV trans illuminator. Be aware that DNA will diffuse within the gel over the time. An examination or photography should be take place shortly after the cessation of electrophoresis. So, different applications of agarose gel electrophoresis: the separation of restriction enzyme digested DNA, including the genomic DNA, prior to Southern Blot transfer, and it is often used for the separating RNA prior to Northern transfer. The analysis of PCR products after the polymerase change reaction to access for the target DNA amplification, allowing the estimation of size DNA molecule using the DNA marker or ladder, which contain the DNA fragment of various known sizes. Allow the rough estimation of the DNA quantity and qualitative. The quantitative is assessed using the lambda DNA ladder, which contain the specific amount of DNA in different band. The quality of DNA is assessed by the observing the absence of streaking or fragment or contaminating DNA band. Next, the other technique rely on agarose gel electrophoresis of the DNA separation, including DNA fingerprinting. So different advantages and disadvantages of agarose gel electrophoresis. The advantages is it can be easily poured and does not denature the sample, and the sample can also recovered. And the different disadvantages that the gel can be melt during the electrophoresis. The buffer can become exhausted, and different form of genetic material may run in the unpredictable form. So please subscribe my channel too, and thank you.